In addition to its roles in DNA replication and gene expression, the nucleus has an important physical impact on cellular behaviour. Keith Burridge and colleagues at the University of North Carolina at Chapel Hill first became interested in the nucleus's physical effects when they identified a mechanosensitive pathway that stiffens the nucleus in response to force, and discovered that perturbing this pathway could in turn alter the cytoskeleton.
There is an older literature saying that perturbations to the nucleus and the nuclear envelope and various proteins involved in connections to the cytoskeleton, the link complex, affects not only the nucleus, but also how cells migrate. And so I talked with Dave at that point and said, what about going back to this old technique of enucleation, which had been pioneered in the 70s, and seeing what happens.
David Graham, a graduate student co-supervised by Burridge and Jim Baer, enucleated cells by centrifuging them at high speeds in the presence of the actin destabilizer cytokalasin, generating cytoplasts that lack their nuclei. Graham first examined how the absence of a nucleus affects cell polarization by plating cytoplasts onto micropatterned surfaces.
So these are geometrically defined shapes of adhesiveness that you can put cells onto, and you can induce a polarity, be it a radial symmetry to the cell, which wouldn't show any sort of polarity, or using a crossbow pattern to show a sort of front-back polarity of the cells. And we use the centrosome, which is always found close to the centroid, or the center of mass of that cell. So when we removed the nucleus, we found the centrosome, much like it was with the nucleus, it was still in the center of the cell. And the Golgi also. That was the other organelle we followed up. Yeah.
Which I think many people would not have expected. And I think they thought the nucleus was very critical in setting up that axis of polarity.
Yeah.
Polarization is important during cell migration, and previous studies have suggested that enucleated cells are still able to move, at least on two-dimensional surfaces.
Our work showed that cytoplasts were able to migrate in just about any scenario in 2D. So they exhibited random migration, and when we put in cues in their environment, cytoplasts were able to directly migrate up a gradient of more matrix protein or towards a higher concentration of chemokine. We also took this old approach called the scratch wound assay. There's such a wealth of literature on the nucleus and its role in polarity in that assay. So we were really surprised to see that cytoplasts could perform in that assay and close this artificial wound state.
However, cytoplasts failed to migrate when they were placed in 3D collagen matrices. In some ways this was a surprise, because a large and bulky nucleus is generally considered to limit the ability of cells to squeeze through small pores in the matrix. But collagen gels are much softer than the glass surfaces used in the 2D migration assays. This seemed to be a crucial difference, because cytoplasts were able to move along one-dimensional lines micropatterned onto rigid glass, an experimental setup that, in most other respects, recapitulates the features of 3D migration.
And then we took them into a 2D environment with different rigidities, going from very soft to much more rigid substrates. And we found things were a little different there. So cytoplasts exhibited a peak cell migration speed on a slightly more rigid substrate than a cell with a nucleus.
This suggested that cells without a nucleus are less responsive to their mechanical environment. This could be explained if cytoplasts have reduced contractility, so that a more rigid substrate is required to activate the mechanosensitive pathways that regulate migration.
One of the big approaches we used was to use traction force microscopy, and we measured the contractile state of cells, and we found that their contractile energy, as well as the traction stress, the energy that they can exert to the substrate that they're adhered to, was lower when we lost the nucleus. We found that they were less able to contract collagen bundles as well. So those are two ways that we could see that the nucleus is important for regulating the overall contractile state or the mechanical state of these cells.
Cells lacking the nuclear lamina protein lamin A, or components of the link complex that connects the nucleus to the cytoskeleton, also showed a reduction in overall contractile energy and a corresponding shift of their peak migration speeds to substrates of higher rigidity. This supports the idea that, by influencing overall cellular contractility, the nucleus is crucial for regulating the cell's response to mechanical cues. Indeed, Graham et al. Think that, through its connections to actin stress fibers and focal adhesions, the nucleus is part of the molecular clutch that transmits traction forces to the cell's substrate.
One of the things that has been puzzling my lab for a long time was we've taken the process of cell migration and broken it down into discrete steps, leading edge protrusion, adhesion, moving the nucleus and all the other cytoplasmic components forward, releasing at the back. But we haven't done a very good job of taking the parts and putting them back together to a whole. And I think Dave's project really brings this point home, that there is mechanical integration across the entire cell. So we're hoping that enucleated cells will really teach us a lot more about how the cell becomes more integrated and pulls together these disparate processes during cell migration.
One of the areas which my lab has been very interested in for a long time is regulation of contraction through the GTPase rho. And so one of the things that Dave is actually looking at the moment is, when you remove the nucleus, do you affect rho signaling? Or could it be one of the other pathways that feed in to regulate myosin? That's one of the things that we're curious about.
For now, though, you can learn more about how enucleated cells reveal differential roles of the nucleus in cell migration, polarity, and mechanotransduction in the paper by Graham et al., published in the March 5, 2018 issue of the Journal of Cell Biology.